Behind auxins, the next of the endogenous hormones to be discovered are known as the gibberellins, or gibberellic acid, GA for short. And they were discovered by some researchers looking at a specific disease in rice. It was in Japan, and so it was called something along the lines of foolish seedling disease. And basically, what was going on is rice is a grass, and rice, as you probably know, is grown in very marsh like or semi flooded conditions. And when it was putting up its flower stalks, which would eventually turn into the rice grains, some of the rice plants started growing unusually tall seed stalks or flower stalks. And they would grow exceedingly tall to where they would fall over into the or onto the moist surface of the ground and rot and basically ruin a portion of the crop. And so they were just looking into what was causing this disease, and they figured out that it was a particular fungus, and the fungus was named Gibberella fujikuri, after the guy who discovered it, but we'll just say Gibberella for now. And that's where the name comes from. They figured out that there was something in the fungus that was causing these cells within the flower stalk to grow abnormally fast or grow abnormally tall anyway. And they isolated this compound, and it was called gibberellic acid. And they came to later find that plants actually produce it themselves as well as this particular fungus producing it. And in fact, all plants produce some version or versions, plural, of gibberellic acid. There are currently no synthetic versions of gibberellic acids, and so while there are some commercially available products that contain higher concentrations of gibberellic acid, it's not very widely used. It's used for a few things in the industry. So let's talk about what some of the responses are to gibberellic acid. Well, first of all, we consider gibberellic acid to be a growth promoter. What are the responses? Well, let's make a list. First, as we illustrated here, or I talked about a second ago, and foremost, is cell elongation. Gibberellic acid stimulates cell elongation. Now, not specifically cell division, but the elongation of the cells themselves. In other words, it stimulates the elasticity of the cell walls, making them more plastic, more elastic, at least temporarily, so that these cells that maybe normally are somewhat of that size would be able to, when expanding their cytoplasm, grow and become longer. And so when you add that up over the thousands of cells in this flower stalk, we get an unusually tall flower stalk. So cell elongation is one response. Another similar response is increased internode length. Okay, I'll just say greater internode length. And an internode is just the distance between two nodes or buds. So we'll just say a node equals a bud. So in other words, an apical meristem, maybe at that point, under higher levels of where it might normally lay down subapicals, something like this, under higher gibberellic acid, there would be a greater distance between the meristems. So that's the internode length versus a normal internode length. Gibberellic acid also, in many plants, stimulates the germination of seeds, which we'll talk in more detail about in coming lectures, but is highly involved in the production of enzymes necessary for that process. And so we'll just say seed germination. They also stimulate flowering or promote flowering in many plants. And what follows flowering, fruit set and growth, or fruit development, you could say. So, all in all, although it's not specifically promoting any kind of cell division, this promotes the cell elongation in the case of stems, and we pretty much think of all of these as being positive growth responses, and so therefore we say that gibberellic acids are a promoter. So we've had auxins that are both promoter and or inhibitor depending on the tissue and the concentration, but gibberellins for the most part are thought to be growth promoting, or thought of as being growth promoters. Which brings us to our next endogenous hormone, which are referred to as cytokinins. And cytokinins have growth promoting effects as well, and they're considered to be promoters. They're produced primarily in root tips, and from which they get translocated throughout different parts of the plant. And they, for the most part, stimulate cell division. The main effect is referred to as cytokinesis, which is a fancy word for saying cell division. So, if they cause cell division, there are a number of things that they are good for within plants. One is they can reverse dormancy. There's a lot of different types of dormancy possible in plants, such as shoot dormancy, bud dormancy, flower dormancy, seed dormancy for that matter. And it plays a role in all of those cases in terms of coming out of that dormancy. And so I'm going to go ahead and write also, it's used commercially in some cases to stimulate seed germination in otherwise dormant seeds, or to break dormancy in some species. It also triggers flower development. Anytime you have a lot of tissue growing in a short period of time involves lots of cell division, and so you can pretty much bet that cytokinins are involved in that process. They're also involved in light responses, specifically growth responses to light and morphological responses to light, such as what we would call leaf differentiation. Also chloroplast distribution within leaf cells. We could say chloroplast arrangement. And what I'm talking about here is the ability of some plants to do the following. If we look at, let's say here's a cell in a plant that is growing in full sun, and I'm going to draw in a few chloroplasts here. Okay. When the sun is very intense, not all plants are up to utilizing that sun very efficiently. And so what they can do is rearrange their chloroplasts, like I have them here, stacked on top of each other. And so in, for example, like I said, full sun, some plants do this to allow some of the sunlight to pass right through the otherwise clear portions of the cell. And so basically this top chloroplast somewhat shades these chloroplasts underneath it. Now in more shady conditions, some plants can adopt by doing the opposite. A little bit smaller cell, but instead of having their chloroplasts stacked, they will spread them out so that they will catch the maximum amount of that sunlight coming in. In other words, it minimizes the amount that actually can pass through the cell and maximizes what gets absorbed, because typically in shady conditions the light that comes in is not as energetic. It's of longer wavelengths, more towards the red end of the spectrum. It doesn't have as much energy in it, and so photosynthetic capacity is reduced. And so one way to try and deal with that is to redistribute chloroplasts within your leaves, if you're a plant that can do this. And that process of rearranging them in either case is thought to be closely linked with cytokinins. And so there's another role for this endogenous hormone.